Here we are. Hello, everybody. I hope you're well. Hope you're enjoying our fabulous Great Exhibition Road Festival. I can take this off. So that's the first thing I'm going to do. I'm going to bore you about face masks for the first minute or so. As you can see, Miranda and I are not wearing one because, well, for one thing, we're separated and we're not by a metre and we're not facing each other. But also we're on stage, so we're separated from you. So that's why we're taking our face masks off, so we're not a danger to you. But we are going to ask you during this session to keep yours on, because some of you are sitting closer together and so on. So we're sticking by the COVID rules, so it does mean we're up here without ours on. But I've explained why. We're not being hypocrites, but we'd like you to leave yours on, if you will. Really appreciate that. So yeah, we've got a few people filtering in, which is lovely. I think the more the merrier, so keep coming in and filling this beautiful room here at the Royal Geographical Society. I'm Gareth Mitchell. I'll be hosting this session. I'm a radio broadcaster and science communication lecturer. And you're about to meet the excellent Miranda, who I'll tell you a little bit more about in the introduction. But just to get us there, I do want to say a massive welcome to our online audience as well. We're on the internet, Miranda. We're streaming, I'm told. We are streaming. So hello out there on the internet. So if you have any questions for Miranda after her talk, then send them in and they will arrive onto my iPad. So I'll be able to pose those to Miranda, and also you in the audience. We're not leaving you out. You'll see there's a microphone at the front there. So when we get into the Q&A, we'll be asking you to come to the microphone to pose your questions. No singing though, please. We had one yesterday. It was terrible. Okay, so that's probably all you need to know, apart from that this talk, as you'll have seen when you walked in, it's part of the One World series. So this is the second of 2 days here in this theatre, where we've had panel discussions and talks on so many different aspects of climate change. We've spoken about eco-anxiety, for instance. We've just had a panel just now about communicating messages around climate change. We've looked at transport. We've looked at food production. So there's a lot to get into. And what I really like about the talk you're about to hear, it's going to kind of bring us up to date. We're going to talk about some modern up-to-date technologies, but Miranda's going to take us back into the history. So I really feel as if this talk is going to gel quite a few of the ideas that we've explored already in this theatre. So no worries, by the way, if you weren't at the previous sessions. We don't mind. You're allowed to only come to one. You've got a life elsewhere. But no worries, because we have recorded them. So they'll be on the greatexhibitionroadfestival.co.uk website for you to watch at your leisure. And I don't know if they're all up there at the moment. It might take them a while to top and tail them and put them up there. But it is worth just going back to this website in coming days to see some of the talks that you might have liked to have got to and you might have missed. So I think that's enough preamble from me, other than to introduce our speaker, who is Miranda Lowe. She's just come fresh from a day in the life session as well. So we're keeping you busy, aren't we, Miranda? There's no time for lunch or anything. She's hop from one session to another. But Miranda is a Principal Curator at the Natural History Museum, which of course is participating in this Great Exhibition Road Festival. And Miranda looks after the museum's coral reef and crustacean collection. And as well as shining a light on the various collection approaches employed by the likes of Darwin and their crews, the collections that Miranda looks after could also reveal the future of these threatened marine environments. Now, during her talk, Miranda's going to investigate the moral arguments over some of the historical methods of collection, as well as to understand how applying modern technologies such as aircraft 3D scanners could then point to how these ecosystems will fare as the world and our oceans warm. So a very warm welcome to you, Miranda. The podium is yours, and over to you.
Thank you. Right.
Yeah.
All right. Okay. Well, welcome everyone. Right. So here we go. We're going to start. So most people, although this is an enhanced, colourful version of it, but coral reefs are generally quite colourful, biodiverse. Known as biodiversity hot spots as well. So that's what coral reefs should really look like. Full of life, lots of fish and other organisms around on various parts of the reef. So, forgive me if some of you already know this already, but I just want to make sure that I'm explaining and that people understand, what is a coral reef. So they're known as, and can be known as the rainforests of the sea. As I said, very diverse. They have the greatest expression of life. Many forms of different types of corals, they're on the reef, and, they're one of the largest ecosystems on the Earth with, 30 of the 34 known animal phyla within there. And that image you can see here that I've got to my right shows the different forms of corals that you can actually find on a reef. So you've got the branching corals, and that kind of species or genus, predominantly that form branching corals that are cosmopolitan, that are found throughout many reefs, can be the kind of coral that's known as the staghorn coral or acropora. So that's a branching form of coral. You've got the, oh gosh, I can't even say that, phaleaceous type of coral, very ornate form, and you can actually see a form of that kind of coral within the museum itself in the Hinsey Hall. It's a turbinarian coral, and so there's one in one of the bays there, collected by William Saville Kent, which I talk about a little bit later on. We've then got a kind of like a builder coral. You can see that circular one there just underneath the branching coral, a massive or lobate coral. And that is a form of commonly known as a brain coral, because it does actually have the formations and designs of similar to the human brain. And so that's a builder coral, kind of normally forming like the basis of a reef, and then all other corals and sponges and other animals stemming off that. You've got just below there the free-living type of coral. So that's a solitary one, and the name escapes me of the genus of that one, so apologies for it. But that's one that lives on its own, because what I would say, that corals are colonial organisms, so they're lots of an animal as well, so they're not plants, they're animals there on the reef. So that's a solitary one at the bottom there. And then you've got things that are laminar and encrusting. So the laminar ones, I like to call them like plate corals, because that's in their design. They're quite thin in terms of their structure. So on the second point there, you can see that I'm saying that, it's composed, coral reefs are composed of animals called stony corals. There are soft corals too that make up parts of the reef, but the stony corals are the predominant, and the basis for the reef formation. Tiny organisms that I mentioned that are colonial, that are polyps. So, for instance, on the branching coral, or when you look at that form of coral, you will see lots of tiny holes there. But predominantly when the flesh has died back, it becomes more exposed, and they are called corallites. But with a living coral, those tiny holes are all the feeding organisms of the coral itself. They're called polyps, and I like to refer to the polyps individually a bit like, if you can imagine, a tiny sea anemone. So it's like tiny holes all over that coral with tentacles kind of sticking out. So they're the mouths, the feeding organisms within a coral, those polyps that look similar to sea anemones. But the skeleton of the corals themselves, when a lot of the flesh and the algae that are living on the corals and the living coral reef, when that dies back, you get exposure of those holes called corallites, and you get exposure of the skeleton of the coral, which is composed of calcium carbonate, similar to chalk limestone. And the polyps, when they live on there, they reproduce, and they have a particular life cycle. So when they die, so they have a larval life cycle. And the interesting thing about corals themselves, that when they are attaching themselves, a bit similar to like barnacles, they produce a secretion that allows them to stick and adhere to themselves, to the shoreline, to other debris as well there. So I've just given you a highlight of the different forms. And for coral reefs themselves, and that's why we're all here today, and I want to impress on you why we should care about the reefs, is that they only thrive in warm, clear waters where you know, the chemistry is suitable for them and the other organisms that live around them. And, you know, pollution is a major issue, and the human impact to the reefs that can cause impact and dieback of the coral reefs themselves. So I mentioned a little bit, but these are the kind of ranges of animals that actually live on the reefs. And so that's why I mentioned they're known as biodiversity hotspots. If those reefs are impacted, then, and they die off, we lose the diversity of species of corals, then we will lose all of these kinds of animals. The shrimps, you know, all the different crustaceans. There's a particular cleaner shrimp that lives on certain coral reefs and is there to actually keep the reef clean. Should a reef get a disease such as white band disease, which would then ravage the reef, and you would lose all the algae and all of the soft flesh, one for a better word, on the reef itself, so the reef will die back, then you'll get exposure of the skeleton. You'll get what looks like there is a bleaching also, but you'll get that look of the reef dying back, that it looks like it's bleached. So that's why we need those cleaner shrimps. They can eat that disease and clean the reef at a rate so that it doesn't spread so fast. So it's important that we have these homes and these spaces for these different species. So we've got sponges, and I've got an image there of different sea stars, the cushion star and a traditional form of starfish there. We've got squid and octopus. Octopus are the, you know, in terms of marine invertebrates, the, you know, most intelligent animal of invertebrates within the ocean as well. And we've got the fish. So in terms of coral reefs, they are worth billions to certain, you know, economies, to the fishing industry as well. So we have to be mindful in terms of fisheries and fishing for the reef, but because also that causes our human impact on the reefs there, but also in terms of the fish using the reefs to be able to hide from predators, but also to rear their young as well. So it increases fish stocks there. But sometimes corals can look like this. I hinted at bleaching and the reef dieback. So what you can see here, these are parts of the museum's collections. So the first top two pictures are from a Reframe project looking at fossil corals. And there are some either side of the central picture at the bottom. So those are fossilized corals within our collections. So we have a Reframe project looking at fossil corals within Indonesia, and so some of that material will remain within our collections for future study, as well as some will be returned. The image in the middle is of what we call recent corals, so corals that are still living in various reefs around the world today, but actually that picture is of some of the historical recent coral material that was collected on an expedition by the ship HMS Challenger. So that was in, got to get my dates right, 1872 to 1876, if I'm correct. And that expedition is considered the birth of oceanographic science. So before those dates, there were other people, such as Darwin, which I will get to, that were doing reef surveys. But for this expedition in particular at that time, there were a lot of scientists and naturalists from around the world that went on board this ship specifically with research collections, such as how deep is the ocean, to find that out. And in terms of the coral specimens in that central picture there at the bottom, out of that collection, there were thousands of new species of coral that were found that were new to science. So from that Challenger expedition alone, not just for the corals, but for other animals that were collected on that, there were at that time in the 1870s described about 4,500 species, and there is still some parts of those collections that haven't been accessed, the jars of them haven't been opened, or the boxes since that time, so there may be more species new to science there. But it's a really rich resource of historical information that we can use today in current research. So this is a young Charles Darwin. He was young once, because a lot of the images we see of him are as of an older man. And so he was a pioneer at his time of reef transects, reef ecology, reef zonation as it's known today, and ancient reef modelling. So in his younger years, in his early 20s, he went on the voyage on a ship called HMS Beagle in the mid-1830s. And on that voyage, he was making lots of observations about lots of animals and things, but in particular, looking at various kinds of reef formations, such as atolls, barrier reefs, and lagoons. And on that expedition, he was forming his own theories and ideas on how that could come about. And what you can see there is an image, and a lot of us are familiarized with the I think diagram that he drew. So this is, I think, about atoll formations. Now, atolls are not the easiest of reefs because you can find them just below the ocean surface or much deeper. And so what he was doing on the Beagle when they went to Cocos Keeling Islands near the Indian Ocean, what he was doing a lot of mapping and survey and reef surveys, and he extensively mapped. I think he did at that point in time the best mapping in the world of the reefs, and it was really important as well to know where various types of reefs are, in particular for atolls, because if they're just below, well, any reef, if they're just below the surface and you've got your ship going across it, it can destroy that ship, and also the ship itself can destroy the reef and vice versa. So that was really important that he was looking at that. And I just want to read a couple of quotes of what Darwin said at that point in time. He said, at Cocos Keeling, I need to, 1, study how reef organisms make reef rock, and 2, satisfy myself that coral reef organisms grow most vigorously in shallow water along reef edges and reef fronts. So, you know, there are extensive letters in his correspondence explaining that's how I'm able to read the quote, and we have that diagram, and he was looking at subsidence as well of the reefs at that point in time, which was really important. He, so Cocos Keeling, he was there in 1835. It wasn't until 1842, several years later after the Beagle expedition, that he actually wrote up a lot of the work that he was doing on reef formation. So there is a publication about that. And you know, it's claimed that Darwin is most famous for his theory on atolls and reef subsidence on the west coast of South America. Well, he quotes, he says, on the west coast of South America, before I had seen a true coral reef, he had come up with that theory, that idea of subsidence. He summarized the coral reefs, that they might provide evidence for the ocean floor subsidence there. And it's thought that he developed his first written and illustrated version of this theory on the Beagle during the time when he was in between Tahiti and New Zealand at that point. So bringing you up to date, actually, and I've only just in the last couple of months been reading around this. So whereas, Darwin's established himself in terms of coral reef formation and the fringing reefs there, and at that point in time, his theories were held in high regard. Only last year, some scientists in France actually have is that the word, debunked, that his ideas on reef modelling, which is interesting because from 1842 to now, you can see how long, science is always evolving, people are always asking questions, and you have to think about it that Darwin was forming these ideas at a time and mapping when there was no satellite navigation. So it was great for his time. Now we have all of that information and equipment that we can use to kind of challenge some of those ideas. So only last year, some scientists in France found some new evidence, and I'm just reading a bit here about this, is that in terms of the atolls, they have found that it is due to the sea levels and cyclic changes in the sea levels that the atolls actually form. And, at that time, Darwin would have had no concept about sea levels going up and down, but, for his time, he did tremendously in the body of work that he did. So I think there's more to come about that in terms of reef formation, and we're looking at reefs all the time in terms of our own impact too, which will give us more information in time of how we should respond, and we should be caring right now about the reef. So I've got some images here of his actual specimens that he collected. There were well over 20. It's a small collection in comparison, but very significant because it was Darwin himself that had collected these at that point in time. You can see the images here around a letter which he wrote, which documents that he collected those in the Beagle. They were donated to the museum at the same time that his work was published in 1842, and they actually were accessioned and registered at the museum in that particular time. And you can't see it on the images here, but they actually have handwritten numbers that links them to the museum. So what he says about the specimens that he collected here, he says, I collected a very interesting series of specimens, beginning with fragments of unaltered coral and ending with others where it is impossible to discover any trace of organic matter. So he's talking about it. And these corals are really important, not only because they were collected about that, at that point in time, but now they can be used to inform other information about the living reefs. Because we now have, I mentioned about satellite technology, but we now have more advanced molecular sequencing. So you now can take a subsample of these specimens and sequence the DNA. You can see that some of the corals actually look a bit mucky and dirty. It's not because we haven't been keeping them in the right conditions and not cleaning them. But what has become apparent and been more interesting and important is that brown material on there is the organic matter, which now scientists are wanting to analyse molecularly now to find out what algae was living on top of. What is that organic matter? Because in some parts of the other historical coral reef collections, a lot of the scientists before when collecting, they have bleached those corals, so not just bleaching from the reefs being impacted by bleaching, but actually physically bleaching, so dipping them into a solution to clear off all that organic matter so that it could expose the corallites that you use. So these kind of little kind of pitted holes that have different patterning that will allow you to identify a coral to species. So there's a bias in the collection from that point of view of a collection looking very pristine. But now it's, well, let's not clean everything for that. Maybe let's just leave half a coral uncleaned and leave the rest with the organic matter, so it could be used for future technologies to understand how corals and coral reefs are formed. The other thing I would say about this material was collected but unidentified to full species since, well, they were collected in 1835, donated to the museum in 1842, but only in the last 10 years, since 2011, that there have been 2 scientists at the museum. One of them is Brian Rosen, who's well known for his work on corals throughout the world, in particular, that has managed to identify these corals to species. So look how long they have been in the collection for them to be identified fully, because we've got more information. And also, there has been more exposure about Darwin's work and his information through the online correspondence projects as well. So everything's to hand online, and looking at his mapping and diagrams to find out where he actually found these corals. So moving on, in terms of historical collections, or what is considered now going into the kind of historical and more recent times of collections, are the Chagos coral collections. So this was a collection, I'm going to go forward a little bit and then go back, that was collected of over 6,000 corals on the Chagos Archipelago on 3 expeditions in the 1970s. One thing I would say, so for those of you that don't know, that you can no longer collect corals in the way that the Victorians have. You can get special permits for, scientific study for collection, but you can't collect in that way as they did before. So corals, a number of them, a number of coral reefs are no-take zones and are CITES listed. So meaning that you shouldn't trade and things like that. So it makes the museum's historical collection even more important for people to come and visit and analyse, because you can't collect in that way. But 3 expeditions there. The leads on that were Charles and Anne Shepherd, and I spelt Warwick wrong, so there's a W missing there, Warwick University. And interestingly, for those that are old enough, such as myself, to remember David Bellamy, who was a botanist, a TV presenter as well, he was on that expedition too. So the collection at that time was to create a coral research centre in Newcastle. It didn't actually happen. In the end, in the mid-1990s, that collection was offered to the Natural History Museum, and so we have all of those corals here now for people to access. But what is brilliant about this particular collection is that, and you can see, it has some of the most pristine corals that were collected, but very important so, because the Chagos Archipelago in 2010, 1st of April, was established as a marine protected area. So a no-take zone for corals. That makes, as I was saying, the corals more important to study. And so Charles and Anne Shepherd are actually have been coming back to analyse this collection in many ways. And one of them is that they are also re-identifying some of the species of coral that they had identified back in the 1970s. So this just shows science is progressing all the time, and the characteristics and the tools that you have to understand a reef and the corals within it. So they are doing that. So there's been what we call re-identification, so renaming of the specimens, but also they have been photographing some of the representative species within. I think there are 96 genera within and lots more species to use as an online resource that you can compare a species of coral within that collection with the same species of what it looks like on the living reef in the Chagos Archipelago. And that is all an online resource with the Chagos Conservation Trust. So collaborating with them and other scientists and scientists at Bristol University through there. So this collection has grown in importance because it's marine, those islands are marine protected area, but also in terms of accessibility to museum collections in general. So, and we have also displayed some of this material in 2015 when we had a coral reef exhibition. I think it was called Corals, Secret Cities Under the Sea. So this is just some information about where the Chagos Archipelago is located in the Indian Ocean, and it's about 70 tiny islands, the biggest of them, Diego Garcia. Right. So what is really important now in terms of what's going to be happening in the next month or so, and what's happening today in terms of discussions about reef and ecosystems and zonations and the human impact on coral reefs, is where you can find all of this information. It's really important in terms of research worldwide for scientists to have access to information. And during the last 2.5 years, we haven't been able to visit our collections to get that information. So online and finding and digital discoveries are even more important. So the museum has a data portal where you can find all of this information. And back in 2004, actually, you can see there's a web page. I created a web page about the Chagos Islands and that coral collection. And there are a number of data sets which are also in the data portal associated with it, because I could see because you can use this information to influence policy as well as research, government policy. And I could see what was happening as well politically. And I thought, right, people need to have access to this information to be, create the right conservation legislation, but also in terms of ordinary members of the public accessing this information too, to understand what corals are about, in particular this marine protected area of the Chagos Archipelago itself. So it's all there to search and available online, and we're continually adding to it as well. And also not just adding from information from within the collections, but when people look at the specimens from other government organisations, they may notice that there's inaccuracy in the location or who collected it or where it was collected from. Sometimes they're just mislabeling or misinterpretation of sometimes the handwritten labels. So we want, and we are creating a verified source for information to influence environment and environmental policy. So one of the many things that have come out of having that collection and collaborating with organisations, in the last couple of years, Zoological Society of London, ZSL, they had a Connect Chagos project. So working with the indigenous people from the Chagos Archipelago. And so there are a number of communities residing in the UK, in Crawley, Derby and Manchester as well. So ZSL had that project engaging with those communities and various kind of training about the coral reefs of their home islands, also teaching them how to dive in the UK and things like that, and how to connect with their other communities that are residing in Mauritius and the Seychelles as well. So that project, I think, was from 2012 to 2015, but you can watch a video, a summary about it. And in more recent times, because of what was going on in the last 18 months of the amplification of Black Lives Matter and people looking at amplifying voices and indigenous heritage of people of colour, the Chagos, there has a group that's been formed last October 2020, called the Chagosian Voices, and they are working with the Chagos Conservation Trust, so mutually both working together, and they had their first conference in December last year as well. So there's a programme of works about amplifying indigenous voices voices, cultural heritage, how that feeds into science and Western science, and culturally, you know, what they understand about the reefs, living, you know, as islanders living amongst the reefs, what that means, as well as, you know, that area being established as a no-take zone and us trying to understand and caring for a living reef now. So talking of, you know, amplification of Indigenous voices and people of colour in terms of the whole, you know, climate change arena, I'm also part of a group that also was put together by an organisation called Doc Society, and this group is called Climate Reframe. And again, it's about amplifying Black, Asian and minority ethnic voices in the UK environmental movement, because me standing up here talking as a scientist and a curator at the museum, that's great as well. But what about my, you know, my cultural heritage? You know, what we do, what all of us do here, does impact other people at the other side of the world, and sometimes those voices are not heard of those islanders that have to live with the impact that, for instance, I use myself as an example, that I might do by just having a mobile phone or anything else like that. And so we all have to be aware of the choices that we make in our everyday lives. So Climate Reframe at the moment consists of 100 Black and Asian people that work in the environmental sector. So it's not only just scientists or curators, but it's people in the media, those that work grassroots with communities in the UK that are really care and conscious about not only the ocean but about the planet in general. But we're looking to extend that membership, and we've been talking about that just only last Friday there. But I urge you all to have a look at what's going on with that group. So what other things do corals tell us about the environment? And what you can see here, probably if many of you have visited the Natural History Museum, passed it hundreds of times, in our lasting impressions gallery, there are 2 corals in an exhibition case there. And as you can see, one of them is sectioned or sliced in half. And that's a coral you can see from the labelling there, Poraites lutea, collected in Phuket in Thailand. And you can see there's a red band running through it. And as it says there, and I've had to update this label, so I'm reading my own writing here. During its lifetime, the sea in which it lived was polluted with iron salt from a factory. The red line shows how the coral absorbed the iron in its chalky skeleton and continued to grow after the pollution had ceased. And it's just showing human impact. So this coral was growing near, in the 1980s, near a tin smelter. Have I got that right? Tin smelter. And there were some scientists from Newcastle University at that point in time that were doing work around there and collecting corals. And it just goes to show, you know, we are creating an impact, because they also said that the reef, when they were doing their observations at that point in time, I think it was 1986 to 1987, they said was going through quite a lot of stress. There were a lot of stress indicators there. So next time you visit the museum, have a look at that. But it's really important that we're mindful, you know, when we want certain products or there's certain industries, you know, where does their runoff go, you know, from making things and goods for us, and the impact that it will have for years, hundreds of years, on the environment, on the coral reef, you know, absorbing that toxicity there, showing that, but you know, for us, it's indicating that impact, so we can see that visually. But we don't want to have many more corals in the collection expressing that, basically, that's what I'm saying. And then unusual 3D scanning documentation that's unraveling the mysteries of the ocean. So on either side, there are what I was talking about earlier, you know, branching corals. So this Acropora corals on either side in the colour photographs. And in the middle, unfortunately, I couldn't find the visual to make it turn, or another coral turn, but that's a 3D image of the corals. And so probably about 5 years ago now, had a PhD student that was doing some studies into reef morphology and how they are created, and does the environment affect the architecture of the reef, how, a bit like the reef design, how it looks. And so one of the beauties of the collection that I look after is that it has a huge time series in certain areas of the same species collected at different points in time. So we were able in a big warehouse, myself and this student's name was Kyle. He was given a handheld 3D scanner that they use in the aviation and engineering industry. It's the first time that the corals have been scanned in that way, or a scanner from that industry had been used on natural history collections to basically scan every coral that we had of a particular species. So we were in this large warehouse, laid everything out on the floor and positioned them, and through various data points were scanning to see what kind of information we could gain from that. And Kyle has taken that information back to, so he was studying here and between an Australian university, and went back there and was doing some living reef research there, and using that, doing some comparative analysis. So that's one way historical collections are currently being used, as I mentioned, from Darwin doing his, on a ship, his just through sight and his ideas, to satellites, DNA research, 3D scanning, there's micro 3D scanning, and who knows what other technologies are to come, because the museum itself are going to be moving some of his collection, some of its collections, 40% of its collections, to a new facility in the next 4 years that will be a science and a science research centre and digitisation centre with new technologies to study our collections and reveal more information on them. So what do we need to protect coral reefs from? I mentioned about white band disease, there's ocean acidification, bleaching, because of temperatures rising, pollution. A lot of the reefs the corals there cannot handle the stress factors of a rise in temperature. They haven't adapted to that in some areas of the world. Overfishing is a big thing, we've got to think about sustainable fishing, the fish that we buy, where we source that from, how much do we need? Do we need to be buying? Because then that does impact, the more we buy, and if we're wasting it, but then the more that you have to fish, the kind of fish that we are sourcing now, that we need to be more aware of, careless tourism impacts on the reef as well. If you're, diving and going on holiday, a lot of people used to, well, not yet, but go to the Red Sea and things like that. Not to touch the reefs in that way, and looking at not just overfishing, but the destructive practices as well, of the gear and how you do it. And there is also predation as well. So I'll give an example of that. So in terms of the Great Barrier Reef in Australia, there's been problems with the crown of thorns starfish just ravishing and just eating across the reef. And that's because of the overfishing. So the fish that would eat the crown of thorns larvae, so the baby starfish as it were, are not present anymore. So then you're getting this population explosion in the crown of thorns starfish. So, we have an impact which then, shifts an ecosystem in a very negative way that then impacts on a reef that won't exist, be less diverse, and impacting on other species that perhaps living on particular reefs or within particular corals because they are species specific. So we have to be mindful of all of that. So what do we need to do? Well, no, why do we need? See, my questions I can't even follow. Why do we need to protect the coral reef? And for all the things that I just just recently explained, to provide a fish nursery so that we've got fish stocks. And other animals have fish to eat as well, breeding grounds for various types of marine animals. And they control the carbon dioxide in the water as well. What can I do to protect coral reefs? And there are many organisations. So this is from NOAA, which gives the tips, which I've explained throughout the talk, about conserving water, choosing sustainable food, volunteering for various conservation trusts and organisations as well. Don't touch. And there are many, many, many other ways that we can get involved. And, in the next month and next couple of weeks, there'll be COP26, where a lot of this information will be amplified. But when all of that finishes, we still got to remind ourselves that, we have to care for our planet. The time is now, the impact is happening now. And we want a future for the reefs and for all of us as human beings and the generations after us to care. And just to think about all of these sustainable goals. There's number 13 in terms of climate action, and number 14, I can't even read that, but that is about what does that say? I can't read. Yes, that's what I thought it said, which I care about as well. But, to care about all of the 17 of those sustainable goals are really important for us and the future of our oceans. So thank you.
Thank you, Miranda, very much indeed for that. So we'll take a few audience questions, but just to get us in the mood for interrogating you. This remarkable, this collection of coral, and to think that Darwin collected it on his voyages, and you have it at your fingertips there in the museum. How much does the collection have left to tell us?
I think a lot more than we actually think. Using think in the same twice in one sentence, because as I explained, because if I'm to take myself back to Charles Darwin's era in his time, I bet they thought, how much more can this tell us at that point? And look what has now, all the new technologies that we have. So I think it's really important to, house and care for these historical collections. In terms of scanning, the detail you can get with different types of scans as well. Not only just the average shape of a coral, but I was explaining about the coral lights within that have specific patterning. So if we could delve deeper into that, to look at that. Molecularly, I just think, the world's literally our oyster of studying just we don't have to take much of a particular coral sample, just scrape the surface or just cut a small, less than a centimetre of it off and grind it up and analyse it and all the data that we can get from it. I just think there's just so much, even what I cannot imagine at this point in time.
Yeah, that's the thing, because there are technologies that haven't been invented yet that will be and can be applied to that collection. It's the gift that keeps on giving, really, isn't it?
Does,
It's, and as for, it's lovely to see handwritten notes by Darwin himself, and of course, some of the curators through the years who've been the custodians of the collection. But was Darwin good at logging all this stuff? Can you read his handwriting okay, or is it all a bit of a mess? Is it okay?
It wasn't that great. Handwriting is a mess, but if you we call it getting your eye in. And if, because I've seen a lot, and the librarians themselves that work with his archives over the years, if you see a lot of that style of handwriting, get to know how they cross their T's and dot their I's, so to speak, there. Or if you know a little bit of background about about the corals, or you've read other information about that reef area, you can then kind of try to understand what he meant in a particular sentence. And also going to other, so I used to go to a lot of conferences, but nothing beats sometimes going behind the scenes in other people's collections and libraries, because I remember I went to Yale, I think it was in 2011, and miraculously they had out on a special display a letter from Charles Darwin to James Dana, who lived in that part of the world, and he was talking to, he's writing, explaining to James Dana about corals, about barnacles, because I also look after Charles Darwin's barnacles as well, that he studied obsessively for 8 years, and within that letter, not only you get scientific information of the two naturalist scientists having conversation about these animals, but also a bit of social history, because Darwin was really ill as well. He was talking about he was really ill when he was studying the barnacles, and he was actually cursing them, but he was persevering. And he's and I know we're talking about corals, but in terms of the barnacles themselves as well, he did a really great work in terms of the classification and the grouping of the barnacles, and a lot of it we still use today. So yeah.
Well, he soldiered on even through illness to keep those specimens coming. And I love that image of the younger Darwin as well. I haven't seen many, if any, pictures of him with, without looking all hipster beardy and rather old or older. Right, audience time. We've got about 5-10 minutes. So Pete's got his hand up. Just see any advance on Pete? Yeah, we've got. Okay. Well, let's start. Chat with the red T-shirt, and then we'll take one from the back, and then Pete. I'm not being horrible to Pete, but he gets questions in other panels as well. So let's see if we can get you all in, and I'll stop waffling and we can get on with it. So yes, if you take the microphone, yes, just come and stand, and then the people at home can hear you.
Yeah, first of all, thank you very much for your amazing and very insightful talk, both in terms of history and in terms of the future. I'm just asking from my background of bioengineering studies, some coral species actually have a very unique molecular as well as structural properties that make them basically the best material for bone implants that you could find. And I was kind of asking, do you think that maybe there could be such thing as a responsible coral harvesting, for example, for this purpose or some other?
They could be. I know they a bit like sponge farming as well, that they are, regions around the world where they're trying to propagate corals and sponges and so on. But when you mentioned about the bones, I forgot, well, there's so many things. So many people I've met and given access to the collections. But there was probably about 10 years ago now, somebody who worked in a hospital or somebody doing research into that thing. So be really interested to see how far, and what you're saying about these studies, linking, because I was interested in the sections and the structure of the corals in terms of bones, and I think eyes as well. I met someone, lenses as well on coral structure. But I'm not kind of up to speed on it, but you've just made me get in the real depths of my mind that I've met some people that I've let them have access to. So yeah, it's great.
Yeah, and just quickly on that point about, if it turned out there were benefits from it, could the corals be harvested sustainably?
Well, I'd like to think so. They'll have to, well, like there always is, a lot of conversation around this politically and regions where there could be. But I think there's, I think that should, why shouldn't it be able to happen? Well, sustainability, this is our whole conversation. We should be gearing towards that, yes.
Totally, yeah. Great, thank you. And thanks for your question as well.
Like you.
So at the back as well, I know you've got to come all the way over to the microphone. It's a bit of a long way. Yes. I know it's a faff, but it does mean that the people at home will hear you. So here we go. It's good for your step count anyway. So.
Thank you very much. And it's so great to hear an appreciation of the historical value of all of these. Is it theoretically possible to go back to some places to get the other parts of the same coral colony that Darwin collected, because they live hundreds of years, or the colony does?
Yeah. It is possible, and there have been people doing that in other areas. I mentioned at the beginning, in terms of our fossil coral collection, that they've been doing some recent collaborative and comparative studies in Indonesia. So nobody specifically has done that project in terms of Darwin's corals, but I'm sure there's been researchers independently in that area. But that's something I will check. But yes, I mean, that's, it's feasible if you get the funding as well to go back and do those studies. But yeah, but it has been done with other parts of our collection. I talk about, in terms of like planktonic samples that were collected in the 1920s and 30s off the coast of the Falkland Islands, and so there are current studies going there, and the British Antarctic Survey have been using the historical samples and identifying the specimens in those, because some of these things are in jars that have never been opened since they were collected, and they've been opened and form part of a current study in that region. So, yeah.
Great, thank you for your questions. We've got one at the back, and then Pete. So I'll get you to if you just come over to the mic, and then I'll get both of you to pose your questions, and we'll see if we've got time. We've only got about 3 minutes, so we'll put Miranda on the spot if she can do about a minute each on these. So let's take one after the other.
Hi, thank you so much. That was really interesting. I was just wondering, what are the kind of practical uses of the 3D modelling of the coral? Yeah. Is there any way that can kind of like support the modern coral conservation?
Okay, so yeah, well, hold that thought. That's a great question. And then Pete, what about yours? So you've got a bit of thinking time now, Miranda. That's handy.
This is just going to be a yes or no answer, so it should be quite easy. I wonder if you agree with my analysis. So we know that burning of fossil fuels have dramatically increased the carbon dioxide in the atmosphere, and that dissolves in the ocean as carbonic acid, and that is the biggest threat to our coral, but it wasn't mentioned in your talk. This is, neither conservation nor the NOAA strategies will save them from acidification. And we know that from the Chatham House report last month that there is an under 5% chance of being under 2 degrees, at which over 99% of the corals are expected to die. So these corals protect against storms and erosion. 500 million people depend on corals for food, protection and income. Nurseries for a wide variety of fish. And if they disappear, we'll have the potential to disrupt the entire ocean ecosystem. Do you agree with that analysis?
Yes.
Yeah. So why wasn't that a part of your talk? That's clearly the most important part of this whole thing.
Yeah, it is. Yeah.
Might be the subject of a different talk in Miranda's defense.
Yes, because I was going from the historical aspect, taking it through, but I agree. Yes, yeah.
Right. So
and I have spoken about that kind of thing before, but I chose this kind of historical aspect there, and leading into, what it can be, the information to use for to amplify people to care about the reefs and the studies there. But yes, those stats, yes. Yeah. I'd agree. Yeah. I've got nothing more to say.
A combination of your talk and Pete's excellently researched question. We had the best of both worlds there, didn't we?
Yeah, so that's right.
Job done, just in time for the end. So what about our friend at the back? So the practical uses of this 3D scanning of coral and where that could go?
Well, I don't do much 3D scanning, but I, lend the corals for particular research, and there's a lot more that can be done. And as I said, the museum is moving towards a research facility that will do that exclusively in a lot more depth and detail. Because in terms of the research that we're doing at the museum at the moment, a lot of the scanning is around the skulls, the bones, the taxidermy as well. As I mentioned, Carl using that scanner from the engineering industry, that was the first time, and that was only like 5 years ago that ever happened. So there needs to be more of that, and uses, the information that it can reveal in terms of that. So it's just, you know, in terms of the collections that I look after, that's a new thing that's just happening, or that people are wanting to transfer that kind of technique to that kind of collection, because it's, as I say, it's been used a lot for skulls and bones in that way.
Okay, well that's great. Well, before I thank you on behalf of us all, Miranda, just a quick reminder about you can catch up with this session if you want to, and others, especially when you sat through a talk. There might be bits that you missed, brilliant though it was. I mean, I'll certainly watch it again, and you always pick things up the second time. And if you'd like to give us feedback on the talk, which we'd appreciate very much, look at that. There's a QR code right there for you. So I'd love to hear what you think of it, and just the festival in general, and follow that link as well. So feedback is really, really handy. And don't forget, stay in touch with us. You've got the URL down there. It's not all over today. It's not just a weekend thing, this festival. So there are events going on right up through the week ahead. And we really hope that you'll take part. You know, the more you take part, the more likely we are to get the funding to get it all together again next year. So we need you folks, we really do. All right, that's my preachy bit over, but I really mean it. But what I also mean is that we're very, very grateful to Miranda for her talk. By the way, when we finish, we're going to have to ask you to leave quite quickly, because we have to wipe everything down, let the aerosol settle, and get another panel in. So don't forget, just grab your stuff and then go as soon as you can that way. But with that said, I keep forgetting to say that, so I've got it in there. Huge thanks to Miranda, huge thanks to our online audience for streaming and watching us as well, but also to you, and also for those great questions we had at the end. Thanks for coming, and thanks for supporting the festival. Enjoy the rest of your day. And thank you, Miranda.
Thank you.
Look at that.